Right now, in the exact second it takes you to read this line, your body has already begun making silent decisions you never consciously approved. Somewhere between your last bite of food and this very moment, a hidden biological chain reaction quietly started rewriting how your cells generate energy. It is deciding how your liver spends its reserves, and strangely, which of its own cells deserve to survive. Over the next 48 hours, this chain does not slow down even slightly, it only builds momentum. By the time you reach the final hour of this story, you will finally understand why scientists obsess over this exact window. Most people assume fasting begins the moment they decide to stop eating, but biology disagrees entirely with that comfortable assumption. Your body runs on its own internal accounting system, one that starts adjusting long before you would ever call yourself fasting. Within roughly 1 hour of finishing a meal, blood glucose peaks sharply and then quietly begins its predictable decline. Tissues either burn that glucose immediately for use or convert it into storage forms for later. Nothing about this feels dramatic, nothing you would notice while sitting calmly at your desk working. Yet this quiet shift is the very first domino in a sequence touching almost every organ system. By the 2-hour mark, something far more decisive begins happening deep inside your bloodstream. Blood glucose returns to its normal baseline, and that single drop signals your pancreas to pull back. Insulin secretion slows dramatically, and insulin is often oversimplified as merely the fat storage hormone. In truth, insulin behaves more like a traffic controller deciding where every unit of glucose travels. When insulin falls, that entire traffic pattern reverses direction almost instantly and without warning. The body stops receiving orders to store energy and quietly begins preparing to release it. This is exactly where your liver enters the unfolding story, and it enters early. As glucose declines and insulin falls further, the liver responds by breaking down its own glycogen reserves. This process, called glycogenesis, releases stored glucose directly back into your bloodstream. Think of glycogen as your body's short-term savings account, built specifically for moments exactly like this. What almost nobody realizes is that this withdrawal begins within hours, not days later. The clock most people imagine starting tomorrow morning actually started quietly before dinner even finished digesting completely. By the 4-hour mark, your body has fully exited what physiologists call the fed state. Fasting, in the true physiological sense, generally begins somewhere between 2 and 4 hours after eating. It is strange to realize you are technically fasting most of every single ordinary day. This happens simply in the gaps between meals, without you ever labeling it that way. Your liver's glycogen reserve is not infinite, and it was never designed to be. Any glucose your body does not immediately use gets stored away as glycogen. Once that food-derived fuel eventually runs out, hormonal responses convert stored glycogen back into usable glucose. Picture this less like a light switch and more like a slowly turning dimmer knob. It is a gradual handoff from freshly eaten energy toward carefully stored reserve energy instead. What makes Hour 4 genuinely fascinating is that your body begins actively rationing rather than reacting. Glucagon, insulin's biological opposite, starts climbing steadily as insulin continues its steady fall. That shifting hormonal ratio is precisely what drives the liver's glycogen release forward. It is not hunger, not willpower, not anything you consciously control or notice. It is a silent hormonal handshake happening entirely beneath your everyday awareness and attention. By this point, your liver has effectively started counting down its own limited reserves.
12 hours in, roughly the length of a normal overnight fast, everything shifts again. Your body crosses into what researchers formally call the basal or postabsorptive state. A person typically enters this state after an overnight fast without any food. By this stage, serum insulin sits low while glucagon continues steadily rising higher. This is the exact metabolic snapshot captured during every routine fasting blood glucose test. It quietly connects a familiar clinical checkup to a much larger biological story. This is also where liver glycogen reserves start running genuinely, seriously low. According to landmark research from physician George Cahill, a foundational figure in starvation metabolism, 12 to 24 hours of fasting typically produces a striking drop in serum glucose. That decline arrives alongside significant depletion of stored hepatic glycogen reserves throughout the liver. A 20% drop might sound modest, almost unremarkable, but consider what depends on it. Your brain requires tightly regulated glucose levels simply to keep functioning normally every second. That is precisely the crossroads where your body must find fuel elsewhere. With glycogen running dangerously low, the body shifts toward an entirely different fuel strategy. Within the first 24 to 48 hours, gluconeogenesis increases dramatically and takes over completely. It draws on amino acids and glycerol to manufacture entirely new glucose supplies. In other words, by hour 12, your body stops merely spending its savings. It begins manufacturing new currency out of raw materials it never planned using. Somewhere around the 16-hour mark, something shifts at the cellular level entirely. This has almost nothing to do with blood sugar and everything to do with individual cells. Inside cells throughout your entire body, a pathway called mTOR begins quietly powering down. As mTOR activity decreases steadily, a different pathway called AMPK begins rising in its place. AMPK behaves almost like an internal alarm system, signaling that resources are becoming genuinely scarce. One of AMPK's most powerful downstream effects is switching on something called autophagy completely. Autophagy is the process where a cell breaks down and recycles its own damaged components. This includes old proteins and worn-out cellular structures that no longer serve their purpose. This is not fringe wellness theory. It is a well-documented cellular maintenance mechanism. It marks a genuine shift from a cell building new material toward pure cleanup mode. Interestingly, this switch does not require anything close to true extreme starvation to begin. Even a moderate, completely ordinary fasting window is enough to tip this internal balance. Some of the clearest evidence comes from animal research rather than direct human trials. Researchers at the Whitehead Institute and MIT found something remarkable in a mouse study. A 24-hour fast prompted intestinal stem cells to shift their metabolism entirely away from carbohydrates. Instead, these cells began burning fat, and this switch measurably improved their regenerative function. This particular finding comes strictly from mice, not confirmed directly in human subjects yet. Still, it offers a vivid, concrete picture of the cellular rewiring likely happening gradually in humans too. The 24-hour mark carries a strange biological contradiction worth pausing to fully understand. While growth signaling pathways quiet down everywhere, one specific hormone does the complete opposite entirely. That hormone is growth hormone, and its fasting behavior has been documented since the 1980s. Researchers tracked growth hormone secretion in healthy men across a normal eating day. They compared it directly against the first day of an extended, carefully monitored water-only fast.
Fasting significantly enhanced growth hormone secretion, amplifying its naturally pulsatile release pattern dramatically. More recent human research echoed this finding with genuinely striking, hard-to-ignore numbers overall. Blood samples taken at the start, at 24 hours, and again at 48 hours confirmed it. They documented marked increases in growth hormone during the first 24 hours of fasting. These changes happened independently of any weight loss, which surprised many researchers studying the pattern. So why would the body boost a growth-promoting hormone while dialing down growth signaling elsewhere? The apparent contradiction resolves once you understand what growth hormone actually accomplishes during fasting. It is not stimulating new tissue construction the way it might during childhood development. Instead, it protects existing lean muscle tissue while mobilizing stored fat for immediate energy use. At hour 24, your body is not confused about whether to build or clean. It is doing something far more sophisticated, safeguarding what already exists while cleanup continues elsewhere. Here is where a popular misconception genuinely deserves correction because it spreads constantly online. Many videos claim that by day 2, your brain runs primarily on ketones already. That is simply not accurate for this specific window of time according to research. In the fed state, your brain relies almost entirely on glucose for fuel constantly. During fasting, it gradually begins co-utilizing ketone bodies alongside glucose, not replacing it outright.
This is gradual co-utilization, not a sudden, dramatic, wholesale metabolic switch happening overnight. The idea that ketones eventually replace glucose comes from a landmark 1967 study. Researchers measured cerebral blood flow in patients undergoing 5 to 6 weeks of continuous starvation. They found ketone bodies had by that point replaced glucose as the brain's dominant fuel. 5 to 6 weeks, not 48 hours, is the actual timeline that research supports. At this current 48-hour stage, ketone levels are genuinely rising, and that part is accurate. But your brain still leans heavily on glucose supplied through ongoing gluconeogenesis from the liver. The dramatic headline claiming your brain now runs entirely on fat belongs to a much later chapter. What is actually happening here is quieter, subtler, and honestly far more interesting than the myth. Your brain is being gradually introduced to an alternative fuel source in small, careful doses. By hour 36, something begins happening to your immune system that almost nobody connects to fasting. Researchers describe this as a kind of temporary demolition happening before eventual reconstruction begins. The clearest early evidence comes again from careful animal research conducted under controlled laboratory conditions. A 24-hour fast in mice increased immune cell counts in the bone marrow specifically. Simultaneously, it reduced those same cells circulating through the blood, spleen, and lymph nodes. This redistribution did not appear caused by cell death, based on proliferation markers researchers measured carefully. Separate research examined monocytes, immune cells produced in bone marrow that fight infection constantly. During fasting, these cells traveled back into the bone marrow to essentially hibernate temporarily. Production of new monocytes simultaneously slowed down, while existing sheltered cells survived noticeably longer overall. This mouse evidence maps onto a broader pattern researchers have started documenting in humans too. That human dimension comes primarily from biogerontologist Walter Longo and his extensive collaborative research team. Their work showed prolonged fasting periods significantly lowered white blood cell counts in participants studied.
In the mouse portion specifically, this shifted stem cell signaling from dormant states into active renewal. This particular human data comes from multi-day fasting protocols, not exactly the 36-hour mark discussed here. What is happening at 36 hours appears to be the opening phase of that pattern. Older, less necessary immune cells begin withdrawing from circulation, setting the stage for renewal ahead. There is one specific worry that surfaces in almost every single fasting conversation people have. Is this process actually eating away at my hard-earned muscle tissue right now? The honest, evidence-based answer at 48 hours is more reassuring than most people expect. Research building on foundational work describes an early phase covering roughly the first 2 days. During this window, the body draws fuel from a mix of fat and protein together. That ratio sits at approximately 70% fat and 30% protein throughout this early phase. That 30% is not nothing and explains why some muscle breakdown is genuinely happening now. But here's the crucial detail that most surface-level content conveniently leaves out entirely. This pattern shifts fairly quickly toward a dedicated, protein-sparing mechanism designed specifically to slow breakdown. The specific pathway responsible is known as the glucose-alanine cycle, sometimes called the Cahill cycle. In this pathway, amino acids from muscle breakdown convert into alanine, traveling straight to the liver. There, alanine helps dispose of nitrogen waste while simultaneously generating new glucose through gluconeogenesis efficiently. It is a genuinely clever recycling system where muscle is not simply sacrificed carelessly for fuel. Instead, it becomes a controlled, limited, temporary raw material supplier for the liver's glucose production. This entire mechanism was specifically designed by evolution to taper off as fat burning increases. By the time 48 hours arrives, none of these individual systems operate in complete isolation anymore. They converge together, each one influencing the others in ways that make this window truly distinct. Liver glycogen, largely exhausted since around hour 18 to 24, has fully handed control over. Gluconeogenesis and rising fat oxidation now drive your metabolism forward with growing intensity. Growth hormone,
elevated since the 24-hour mark, continues protecting lean tissue while fat mobilization intensifies further. The protein-sparing mechanism described earlier has now had a full day to fully establish itself. This is also the exact point where the phase model marks a genuine metabolic transition forward. The early phase, characterized by that initial 70-30 fuel mix, gives way to something new. A second phase emerges where the fuel mix shifts decisively toward burning stored lipids instead. 48 hours, in other words, is not some arbitrary point randomly marked on a calendar. It sits close to the actual biological hinge, separating two genuinely distinct metabolic states entirely. What makes this moment worth pausing on is how unremarkable it looks from the outside. Someone at hour 48 of a fast does not look noticeably different to anyone watching. They may not even feel dramatically different, depending entirely on their individual body and circumstances. But internally, their liver has completely switched fuel production strategies without any external warning whatsoever. Their hormone profile has shifted in ways that would seem contradictory without this exact context. Their muscle tissue has activated a genuine protective mechanism working quietly behind the scenes constantly. And the earliest signals of immune cell reorganization are already quietly underway beneath the surface. 48 hours turns out to be less a single event and more a convergence point.
Several independent biological clocks briefly line up together in a moment of quiet coordination. Stepping back from all this hour-by-hour detail, a much bigger question deserves genuine consideration here. Why does the human body possess any of these elaborate mechanisms in the first place? The answer connects to something anthropologists have pointed toward across enormous stretches of human history. For the overwhelming majority of that history, reliable daily meals were never actually guaranteed. Periods without food, sometimes lasting several days, were a routine feature of ordinary life. The systems walked through here, glycogen mobilization, the alanine cycle, growth hormone elevation, immune reorganization, are not malfunctions. They represent a coordinated, evolved toolkit refined over an enormous span of time. This toolkit exists specifically for keeping your brain and body functioning through exactly this kind of gap. What is striking, looking back at everything covered, is how little resembles the oversimplified popular version. There is no single dramatic switch flipping suddenly at hour 16, 24, or 48. Instead, there is a layered, overlapping sequence, hormonal shifts here, cellular signaling changes there, unfolding gradually. Each piece has been calibrated by decades of research, spanning foundational starvation studies through modern trials. Perhaps the most grounding takeaway from all of this is remarkably simple and reassuring. Your body is not struggling or improvising during these 48 hours of fasting at all. It is executing a plan, tested, refined, and carried forward across an almost unimaginable stretch of history. The specific checkpoints, hour 4, hour 12, hour 24, hour 36, hour 48, are simply markers. They are checkpoints along one single, continuous, remarkably well-organized biological story unfolding inside you constantly. And that story is far more intricate, more surprising, and more scientifically fascinating than most people ever realize.